Okay, so let's go now to specific to neuronal cell structure. Okay. So this is how it looks like under microscope, yeah, where the structure is already made clearer using fluorescent quantum dots, yeah, using fluorescent quantum dots that when it is being lighted by through the microscope lighting, it will fluoresce. So this shows the cell body, yeah, the structure, and this shows the dendrites. You see here the size of one individual neuron cells is about 10 micron, several 10 micrometers long. This is the photograph or photomicrograph of nerve cell from the hippocampus, the major region of the brain. A voluminous cell body can be recognized with branches called the dendrites. Here, the microscopic structure is visualized by immunoprecipitation with fluorescent quantum dot. So numerous short cellular extensions are called as the dendrites. So this is because they have a tree-like structure, tree-like shapes, and this provides the receptive area for the incoming signal from the surrounding neurons. Yeah, as we have discussed in the previous session, and these tree-like dendrites will receive all the neurotransmitter coming out from the surrounding neurons, and which will then open some channels using the transmitter-gated mechanism, as well as by the potential difference, some voltage-gated channel will also be opened. So the receptive area serves as a cellular input for graded inputs from other neurons over the neuronal synapses, or from physical stimulus, direct impact, in this case when it is a sensory neuron, or even from a non-neuronal receptor cell, over synapses. Or if it is in the central nervous system, then the input will be captured by this receptive area, the dendrites, from the surrounding neurons. So later, when you are in semester 7, you will learn artificial intelligence subject, where it is an algorithm which is based on the mimic of the neural structure. Yeah. One of the algorithm in artificial intelligence is called as the artificial neuronal network. A membrane imbalance UR, known as the electric gradient potential, is generated in this receptive area in response to any of the cellular inputs. So after that, this input will be propagated to the cell body known as the soma, which is the metabolic center of that cell. Then a single long tubular extension could be from 100 micrometer up to 3 meter in length, yeah, depending on the position in the body. This known as the action will conduct the non-graded action potential, all or none events. At the end of the action, a presynaptic terminal resides, which releases a chemical transmitter in a graded manner through in quantum steps, so in graded manner. So similar like this one, yeah, the transmitter will be released based on the potential across this action. And then the transmitter release will serve as a cellular output towards the other neurons or the muscular cell. If it is motoric neuron, if this neuron is connected to a muscle cell, then it is a motoric neuron. So this is the neuron's structure, similar as what we have seen before, actually. So this is the dendrites. The dendrites act like tree-like shape structure with free sensorial endings. So this is the trigger zone. So the dendrites receive, depending on how many connections, it could receive from one single other neuron or three other single neurons, or from single muscular cell, for example. So this is, it will collect the input from the branches, and then it will trigger. This is the trigger zone where it accumulate, and then when it is above certain threshold, it will create a response propagated through this action and until the ends, yeah, the neuronal synapse on the dendrite here. It could branch into a single, or in this case, in this picture, it is two. The output of this neuronal cell is propagated or transmitted to two other neurons. Okay, the association neurons. B here is when that sensory neuron is connected to a secondary receptor cell. Yeah. This is a hair cell. Hair cell is usually located in your ear, yeah, where it has this hair bundle, which then upon mechanical deflection due to the vibration of the membrane inside the tympanic membrane inside our ear, the hair bundle will deflect, and when upon deflection, it will open the voltage ion channel, the ion channel, voltage gated ion channel. Then ion will be collected by this hair cell, and then the transmitter will be released to this trigger zone of the next sensory neuron, and the response will be propagated through the myelinated axon into the branch axons towards our central nervous system. Okay. So if we model them like biomedical and artificial biomedical instrumentation, then the signal coming from the synapse, yeah, into the dendrites can be modeled as a graded or as a analog signal. Goes to the trigger zone, which will act as an analog to digital converter. Yeah, remember in the introduction to biomedical engineering about ADC, analog to digital converter. Then it will create either 1 or 0, yeah, all or none signal, so digital signal propagated through these neurons, and then in the synapse it will be then converted to analog signal again in the form of transmitter release, yeah, to the surrounding associative neurons. So the sensory neurons, there are three types of neurons: sensory neurons, association neurons or the interneurons, and the motor neurons. The sensory neurons or the afferent, yeah, afferent neurons includes or interconnected with the sensory receptors located in the body or body's periphery, in the skins, in the ears, in the nose, in the tongue, etc. So the sensory neurons will convey the sensory informations toward the central nervous system, our brain, and also the nervous system in our back. Namely, sensory inputs such as light, sound, pressure, chemicals, or heat will activate the corresponding receptors on the cellular level, like say the activated gated channel for ions in the membrane of the dendrites. So the resulting graded output of the receptor is converted into all or none action potential, 1 or 0, okay, which then propagate along the axon towards the neuronal synapses as communication units with other neurons. Arriving in the central nervous system, an appropriate response is provoked, for example, as a conscious perception or an involuntary reflection. Involuntary reflection means when the synapse is short-circuiting the central nervous system of the sensory neuron, directly goes into the motor neuron. And then inside, trigger to the muscle cell directly. But if it goes from the to the central nervous system, it will create conscious perception in our brain, and then we decide. So that is when we decide what to move, how to move, etc. Yeah. Association neurons or interneurons interconnect other neurons via synapses and ensure functional integrity of the CNS, central nervous system. So the association neurons is the largest class of neuron, the class which has the largest number of cells inside our body, including in our brain, of course. Then the motoric neuron is called as the afferent neuron. Yeah. Sensory is afferent, motoric neuron is afferent neurons. This will conduct the actions potential from the central nervous system toward the effector organs, such as the muscle cell when we want to move, or glands. Glands means when like the salivary gland to excite more saliva, sweat gland to excite more sweat, etc. Namely, series of action potentials are converted into a graded release of neurotransmitter. Yeah, usually it is in the form of acetylcholine or in the neuromuscular synapse that triggers muscular contraction. So yeah, regarding acetylcholine, because we mentioned this many times, yeah. So many of the what do you call it, prohibited drugs like narcotics, etc. They consist of these neurotransmitter molecules. So when people drink it or smoke it, yeah, usually they will this they can nicotines like that, yeah, including nicotines in the usual smoke. They can bind easily to the nervous system as this neurotransmitter. That's why then some people will get this sensation of being in what hallucination state, or in something like in the calm state, but not really calm, but actually, but being in a hallucination state. Yeah. So that is why also that creates some addiction, yeah, in the people who use these drugs, and it is usually that is also why it is dangerous. So it is not good to use these narcotics based drugs if you use it without proper doctor prescription. Although some of these drugs are used as a painkiller to reduce the effect of pain, yeah, in after surgery or something like that, because it blocks or it incite different kind of neuronal response to cheat the brain that you are actually not feeling the pain, something like that. Okay, so this is how neuron function works, the signal that occurs. So as we know, actually the digital signal also works inside our body, not only in our artificial engineered instrumentation. So like this, for example, when the amplitude, this is when the sensor neuron, in the case of sensor neuron, attached to a muscle. So when the muscle is being stretched and the stretch period is like this, the amplitude of the stretching is small, yeah, then you see that it is still below the threshold of that sensory neuron. So it will not trigger a response of that sensory neuron. So the action potential propagated will be zero, okay, because it is below the threshold level at the trigger zone. Yeah, the trigger zone, this part. Okay. So, and then when the amplitude becomes higher, yeah, then it will be, this response will be above that threshold of that sensory neuron. And during that period where it is above that threshold, it will trigger this kind of action potential, yeah, this impulses or pulses, yeah, pulses, very sharp pulses, yeah, which has a frequency or period of 1 over FF, yeah. Okay, the period of which depends on the amplitude. So for example, if the amplitude is higher, the amplitude is larger, the response, sorry, the stimuli amplitude, let's say it could be stretching in your skin, for example, it is larger, then it will create more of these pulses. When the amplitude is when the duration of the stretching is longer, also this will create more, yeah, create more number of these pulses. When it is larger, the frequency will be, when the amplitude is larger, the frequency of the amplitude will be higher. It means it creates in one single duration of time more pulses. When the duration of the stretching is longer, of course, then the number will be also more, like this. Yeah, this is the, so this is called the endotonic phasic receptor. When in the phasic receptor, on the other hand, this will create, the stretching will create these two big stimuli, and then each will create a series of impulses. So then the propagated stimuli will be in this kind of two series of impulses. So there are 4 elementary types of information that are coded by this sensory neuron. Yeah. First is the stimulus strength. This is the amplitude. Yeah, as we discussed here, the stimulus strength. How high is the stretching? The amplitude. So this is, and the temporal changes of the strength is coded by the frequency of the action potential. So the higher the stretching, so here you see that the more of these pulses, the frequency will be higher. The more of these pulses will be produced or will be outputted for every unit time. So the frequency is higher. The stimulus duration is coded by the number of action potential. So here, this is longer than this one. So it creates more pulses compared to this one. This only creates 4. This, I don't know, more, much more, maybe more than 10. Then the stimulus type. Mechanical, chemical, optical is coded by the type of the receptor, distinct receptor. So mechanical sensory neuron will be different with chemical sensory neuron, and will be different with optical sensory neuron. So the distinct receptor responding only to a particular stimulus type and by distinct neural pathways that carry the relevant action potential towards the central nervous system. So from which position the CNS, the central nervous system, know, oh, this is the response coming from optics, from the eye. This one coming from the tongue, chemical. This one coming from the skin, mechanical action. Then the stimulus location and its spatial dimension provided a topographically distributed receptor. For example, in our finger, it is this. These are one position within our body which has a lot of sensory neurons. That's why it's sensitive towards touching, for example. At which part we know whether the touching at the end of the finger, at this part of the finger, or at the beginning of the finger, etc. Okay? So this is the spatial dimension encoding is due to the topographically distributed receptors across this finger. But when it is located in the hand, for example, we are not that topographically sensitive anymore. We just know that it is in the hand, which part of the hand. Sometimes we are not that sensitive because less receptors are being installed there. Another part which has lot density, a high density of receptor is, of course, in our tongue. Then the receptors can be subdivided into 3 types according to the adaptation speed to a constant excitatory stimulus. Yeah, constant this one, I like this, constant excitatory stimulus. The first is the phasic receptor. Yeah, this one, the phasic receptor. This is the rapidly adapting receptor, which is silent when a constant stimulus occurs. So when this is constant, yeah, after a while it is being here at the beginning. When it is increased, suddenly increased, it excite. Yeah, it excite the stimuli. Yeah, there is excitation of the sensory neuron. But after a while, it immediately goes zero, even though the stretching still occurs. Understand? So when the stretching of the skin, for example, finish, the skin finish, then excite again. Yeah, so to indicate to the central nervous system that something stops stretching the skin like this, but then immediately goes down. So this first strain of impulses indicate when stretching occurs. The other ones indicate when the stretching finishes or stops. If the stimulation persists, its perception gradually fades from consciousness. The receptor fires only when the stimulus intensity increases or decreases. So when it is constant, the central nervous system doesn't feel the stimulus anymore. The corresponding receptor potentials last only for a short time, independent of the stimulus duration. Like this, even though the stimulus is still happening, it just finishes. This excitation finishes immediately. Okay, clear, everyone? So example are the olfactory sensory neurons. which detects only the change in sense, and the mechanoreceptor in the skin, which respond to the establishment of skin contact or to skin vibration rather than to a continuous skin contact. So for example, you wear your clothes today, right? When you wear your clothing, you feel like you are wearing. But after a while, you forget that you are wearing your clothes. You feel like that? Because you feel already comfortable with it. The feeling comfortable means quantitatively that your neurons doesn't feel anymore that close. Because your mechanoreceptor in the skin basically is a phasic receptor. It only detects when the clothes starts to contact the skin, and when you release the clothes from your skin, it will create another response again. When afterward it's zero. Like now, you feel comfortable because you don't feel a cloth is touching your skin. You feel again when you, for example, put another clothes, thicker clothes, for example, a jacket, something, and you feel. But after a while, when you then start to zero, you don't really feel anymore, except when you are, especially when you are not moving at all. Okay, but when you move, of course, there will be friction between the clothing and your skin, and your skin give this stimuli to your brain that something is above or in contact with your skin. Understood? So this is how our sensors work, how our sensors in our body works. Yeah. So it is the phasic receptor, similar with the olfactory. Yeah, our nose. Usually, our nose are sensitive at the first time when that perfume or whatever touch it, and then we say, "Oh, I smell something." But after a while, our system gets used to that smell, and we don't smell it anymore. Yeah, you experience like this, or you always smell like that? No, right? Any feedback? Any experience? Similarly, of course, not only perfume, you smell something from in the toilet, for example, right? At the beginning, when you enter the toilet, you smell it. But after a while, you stay inside the toilet, you don't smell it anymore. You get used to it because your nose function as a phasic receptor. The receptor, the sensory neurons in your nose functioning as a phasic receptor. When you're going out from that toilet, now you feel fresher. Then you get used to it again. So that is the term getting used to something, to the clothing, to the smell, etc. is because we are using our phasic receptor. Then the tonic phasic receptor, the second part, the slowly adapting receptor. So basically, this part is the tonic phasic receptor. This is a slowly adapting receptor, which yield a train of decelerating action potential at the constant stimulus. So when at the constant stimulus like this, stretch, initially because this is very high amplitude, it creates very high frequency impulses, but slowly the pulse frequency will decrease, and you see that it is getting more sparser between the pulses. The corresponding receptor potential weakens slightly during the stimulus. An example given by the muscle spindle to record the actual muscle length. Yeah, the actual muscle length. Tonic receptor. So very slowly adapting receptor, which yield a continuous strain of action potential with a constant instantaneous frequency at a constant stimulus. The receptor gives information on the stimulus level throughout sustained and prolonged stimulation. So in tonic receptor, it stays. So similar like the common analog sensor, yeah, the tonic receptor. For instance, the baroreceptor in certain arterial vessels, yeah, to monitor the blood pressure. Continuously monitor the blood pressure and its changes, yeah, so that then it can give signal to the nervous system and then to the heart to pump the appropriate pressure to the blood vessels. Okay, so far understood? Any questions? Or sleeping, everyone? Are you awake?
Yes, sir.
Yes.
Sir, can you give an example for the tonic phasic receptor?
Yeah, tonic phasic receptor here, usually the muscle that is located at the end joint. So for example, our biceps is located, the ends of the biceps located in the elbow, yeah, and then the other end at the shoulder, right? So when we move our bicep, we feel when it is contracting or when it is not contracting. Okay, but even though that feeling slightly fades away, but not as quick as the skin contact receptor or the nose receptor. Okay, so because it measures the length. So you are an athlete, right? Yes, but you're more of basketball, yeah? So you don't do strength exercise. Maybe Andy do strength exercise. If you go to the gym, well, I've never done it. Well, let's say when you lift something using your bicep, you feel it, yeah. You even if you hold it, you feel that bicep contraction for some period of time, right? That the perception of the bicep being contracting doesn't fade away easily. Even though if it is too long, then yes, it's getting used to it. Then you release it, you feel it. So because it measure recording the actual muscle length. Yeah, this is tonic phasic receptor. If you compare in contrast with the skin contact, yeah. For example, let's say you touch your hand now with your finger on from the other hand. Then you feel when it starts to contact, right? But after a while, you just if you completely not moving, you feel that sensation of contact. You consider it as you get used to it. You feel it again when your finger is being released from the skin, from your hand skin. So this is the phasic receptor. The phasic receptor here only detects the on and the off phase change. That's why it's called phasic receptor. The tonic phasic receptor, like in the biceps, at the ends of the muscle, they measure this pulse when the amplitude is high. The frequency of the impulse is also high, but then it is what we call it, the frequency gets lower and lower, but it's still there. It is still there, so we still feel, and only after the stretching is turned off, then it completely turned off. So tonic phasic receptor we still feel. In completely tonic receptor, like in the receptor inside our body, the baroreceptor here, measuring the blood pressure all the time. It's really measure the actual level at that particular time. Yeah. So this is how the different neurons, sensory neurons, focus in our body. So in neurons function, we know either excitatory or inhibitory function. In excitatory function, the particular signal will amplify the signal. Particular signal will be amplified so that then it will get excited easily in the trigger zone. Inhibitory, on the other hand, that it create a signal that will inhibit the signal from the other synapse. For example, here, when this is excitatory synapse, which will create a higher positive potential. This is excitatory also positive potential that will work towards the excitation at the trigger zones that will be propagated throughout the action. On the other hand, if there is inhibitory synapse, it will create here negative potential that will, so one of the positive charge here, instead of going to the trigger zone, it will be captured here. So that it will be, so this potential, this voltage here, this field here will be reduced compared to the field coming from the synapse and the synapse. Yeah, this is thicker, right? It is drawn thicker here. Well, this one is drawn thinner because some part of this excitation for a potential is being attracted or being captured by this inhibitory synapse. We call it inhibitory shunt. This is finally induced extracellular compare. So yeah, of course, these are all formed by the training throughout our growing, yeah, from our children and our until now, all creates different excitatory and inhibitory synapse connection within our brain. So here, for example, in the presynaptic action potential, when it is excitatory, in the it will be amplified, yeah, like this, it will create a signal. When it is inhibitory, this action potential will be suppressed. Then it will not create any trigger potential. Something like that. Okay, any questions so far? So about neuron muscle, neuron cells. If not, we go further towards. Wait, sorry.